Today we will focus on using this workflow to identify genomic variation in a biopsy sample, but keep in mind that this workflow applies to not only cancer, but also hereditary disease, rare disease, plus more. Cancer genomes are incredibly diverse and complex. They often have a higher frequency of genetic mutation compared to inherited. Tumors may contain extra or missing genetic material deviating from a multiple of the haploid set. Therefore, informatics pipelines developed around 2N do not work as well for oncology samples. Since the biopsy sample may be a heterogeneous mixture of tumor clones and normal cells, variants may be present at a low variant allele frequency. It is essential to know the tumor percentage of a sample before genomic interrogation. Tumors vary in size, so the amount of sample available for processing may be small and limited. Since genetic mutations in cancer span from a single base pair change to insertions and deletions, copy number changes and rearrangements, a multimodal approach for testing is implemented. One may examine a sample for immunohistochemistry and FISH on the cellular level using slides, and on a molecular level with sequencing and qPCR. Single base pair changes and small indels encompass almost 3/4 of all actionable gene alterations. Looking at our sample, immunohistochemistry for PD-L1 identified expression in at least 50% of the tumor cells. FISH was negative for an ELK-EML4 rearrangement as well as a ROS1 fusion. Sanger sequencing was negative for the hotspot variant in exon 19 of EGFR. Based on these results, the pathologist identified the biopsy as a non-squamous, non-small cell lung carcinoma. Without molecular profiling, this biopsy story would stop here. But what if there was more information that this biopsy can tell us? Could it have a mutation, a rearrangement, or a copy number change? One could perform tests to look at each of these possibilities, but this consumes more of the precious sample, takes time, and costs money for each test. Next-generation sequencing, or NGS, can streamline this process. Next-generation sequencing is also known as massively parallel sequencing because nucleic acid fragments are sequenced at the same time in a parallel manner. To perform next-generation sequencing, nucleic acids are cut into smaller pieces of about 200 base pairs. The jagged ends are repaired so that both strands end at the same point. An adenosine base is added to the blunt end so that a sequencing adapter, index, and barcode can be added to the fragment. Adding unique indexes to a sample identifies it throughout the process so that many samples can be sequenced together. Once the pool of libraries is added to the sequencer, the adapters bind to the sequencing flow cell, creating a bridge. These fragments are amplified to produce many copies, resulting in a cluster. A single fluorescent base is added with each synthesis, and the unbound nucleotides are washed away. After the sequencer captures the image, the dye is washed away and the cycle is repeated. This process continues until the bases of the fragment have been sequenced. Agilent offers a complete NGS workflow for the Illumina sequencers, starting with extracted nucleic acids. This diagram outlines a basic DNA-seq NGS workflow from extracted DNA, fragmentation, library preparation, and target enrichment. Also important in this process is QC. Agilent has many different instrument options for QC, from single samples to ultra-high throughput. Our ELISA platform provides a simplified, customizable informatics pipeline to process sequencing data into a final filtered variant report. We will be taking our biopsy sample throughout this workflow, including QC performed on the 4150 tape station and NGS library preparation using the Magnus NGS prep system. A typical NGS workflow requires 2 laboratory spaces. 1, a pre-PCR amplification area to perform nucleic acid extraction and fragmentation, and 2, a post-PCR amplification area for the Magnus NGS prep system, QC instrumentation, and the NGS sequencer. Agilent optimized this workflow to be compatible with high-quality blood and low-quality DNA. Because of this, it is important to properly QC your sample not only for concentration, but also for quality, so that you do not waste time, money, and resources and can optimize your workflow. The TapeStation family of instruments has a genomic DNA screen tape that can assess the quality of a sample to adjust the input of the sample into the workflow for better performance. As you can see here in the table, a DIN of greater than 8 provides a 10 nanogram to 200 nanogram DNA input. However, as the sample quality decreases, the amount of minimum input has been increased. We examined our biopsy sample using the 4150 TapeStation's genomic screen tape with 3 other samples of varying degradation, including a high-quality HapMap sample NA18507. This slide shows the gel image with the DNA integrity number and the electropherogram overlay. What regions are important for us to look at for lung cancer research? Agilent Sureselect technology can pull out those regions from the overall pool of DNA using streptavidin-tagged complementary RNA probes. The Sureselect Cancer All-in-One Lung Assay consolidates the detection of all variant types into one workflow with a single DNA input. This assay includes all somatic variants associated with non-small cell lung cancer in 20 genes based on the guidelines of the National Comprehensive Cancer Network, the College of American Pathologists, and the European Society for Medical Oncology. It can identify rearrangements in ELK, ROS1, RET, FGFR3, and NTRK1, and copy number changes in ERBB2, CD247, MET1, FGFR1, and PTEN. To overcome the limitations for CNV detection using NGS from somatic samples, we have created a proprietary bait design strategy to place CNV-specific baits near genes of interest. B-allele frequency modeling is used to calculate clonal fraction, which, combined with read depth information, gives sensitive somatic CNV callings. This data from a high-quality cell line and an FFPE-derived sample prepared with the Sureselect Cancer All-in-One Lung Assay shows approximately 98% overall concordance between ERBB2 copy number detection at various tumor fractions. The All-in-One assay includes a probe design targeting recurring breakpoints in genes of interest, coupled with an algorithmic approach utilizing multiple lines of evidence to detect rearrangements. Here we show how translocation breakpoints may be better characterized using NGS than FISH, since breakpoint locations can be identified at the base pair level. Multiple NGS probes target the same region compared to a single FISH probe. This data shows approximately 96% overall concordance in identifying an ELK rearrangement at various variant allele frequencies using Agilent Sureselect Cancer All-in-One Lung Assay on a high-quality cell line and an FFPE-derived sample. Now that we have our extracted DNA sample that has been QC'd, we need to break the genomic DNA into smaller fragments of 150 to 200 base pairs. 2 methods exist to perform this task. The first uses sound waves to break the DNA into bite-sized pieces. This method requires the purchase of a specific instrument, multiple protocols for validation, and sample loss from transferring samples between tubes. The second type is enzymatic fragmentation, in which enzymes shear the larger fragments into smaller pieces. No additional instrumentation is required, and it is compatible with various sample types and inputs. Since it is an enzyme, it minimizes damage to the DNA, and in our method, it can go directly from enzymatic fragmentation into the library preparation without any cleanup. Let's look into the steps of Agilent Sureselect Xths workflow. After the sample has been fragmented to 150 to 200 base pairs, each fragment undergoes end repair and A-tailing, in which the fragments become blunt-ended and an adenosine base is added to the 3' end of each strand. Adapters, which are short DNA oligos containing the primer site for the sequencer, and sample indexes are ligated onto the fragments. A magnetic bead wash is performed for size selection. Finally, the pre-capture library is amplified by PCR. For target enrichment and capture, the pre-capture library is incubated with capture probes, also known as baits, for those regions of interest. Each of these baits contains a streptavidin tag, which is used to bind the fragment of interest to streptavidin beads for separation. Once the regions of interest have been pulled out, they are amplified in a post-capture PCR, resulting in a targeted NGS library ready for sequencing. The Magnus NGS prep system automates the Sureselect Xths workflow from fragmented DNA to the targeted NGS library. The instrument includes all of the necessary equipment to perform NGS in a laboratory. Only 5 minutes of hands-on time is needed when setting up the instrument run and loading reagents and consumables onto the instrument's deck. Using a predefined protocol, we created libraries from our biopsy sample along with 7 other reactions in less than 9 hours, with no user intervention except loading the instrument. The Magnus reagents come pre-aliquoted and can accommodate catalog or custom baits, which makes this a perfect complement for the Sureselect Cancer All-in-One Lung assay. The 4150 tape station processed our lung biopsy sample and the other 3 samples on the high-sensitivity D1000 screen tape. The electropharograms show the final library traces with the expected maximum peak size between 200 and 400 base pairs. After enough samples are processed to fill up the sequencer's capacity, the samples are normalized based on the final library's molarity from the 4150 tape station. Each sample is normalized to 4 nanomolar, and equal volumes of each sample are combined to create the sequencing pool, diluted to the specific molarity required by the sequencer, and control phi-X added. The sequencer is set up based on sequencing recommendations and SOP. Now that our biopsy sample is sequenced, Casey will continue its journey through the informatics pipeline.
Thank you, Bridget. Now that we've walked through the pre-sequencing workflow steps, let's talk about what happens after sequencing. This is specifically the data analysis and interpretation steps. We break this down into 3 layers where we talk about primary analysis, secondary analysis, and tertiary analysis. Primary analysis really includes the sequencing QC metrics and the reads that are produced by our sequencer. Secondary analysis is when we take the sequencing reads and we will then align and call the variants that were identified through our sequencing experiment. The tertiary analysis steps take the variants that we have from secondary analysis and we review these variants in context of relevant databases and relevant papers that we then want to report out on, where we'll draft reports and highlight our results. Let's talk about the primary analysis. In this table, we have some of the standard sequencing QC metrics that will be returned from SAV as part of a primary analysis. Let's look at the right side of this table, which provides some information about our run in context of the phi-X spike-in. The first part of this table is the percent aligned, which tells us the percent of reads that align to the phi-X control genome. This really tells us did the sequencing control behave as expected. Ideally, the percent aligned in this table should approximately match the percent spike-in that we calculated during the loading of our sample into the sequencer. The second component that we compare to the phi-X spike-in is our error rate. These are the number of errors compared to the aligned phi-X control sample. This is done for the entire run, but it also is done for cycles at the 35th cycle and the 75th cycle, where it tells us the error rate for cycles 1 through 35 and 1 through 75 to show whether or not our error rate was higher at the end of the run compared to the beginning portion of the run. The left side of the table provides us more information about the QC metrics for our sequencing run. The first is the percent of Q scores that are greater than 30. It tells us the percent of bases whose quality score is greater than 30, which is a measurement of high quality or not, corresponding to a confidence rate of 99.9% or greater. The next one is going to be the cluster count, both for the raw and the pass-fail. This tells us how well we loaded our sample onto the sequencer. If you over or underloaded, your cluster count PF will probably deviate high from your raw cluster count. The last one of these is the indexing QC metric, which is the percent of reads represented by each sample. When we are pooling our samples, we can calculate the relative proportion of our pooled samples, and we then compare this with our indexing QC to tell us how well we pulled our samples and how each sample performed. We'll now go into the secondary analysis, and Agilent has a solution to assist you with your secondary analysis. This is Alyssa Align and Call, and we're using this for AIO. This is a straightforward alignment and call for all Agilent kits, and it provides extensive QC reports that will be saved for each analysis. The QC information that's provided as part of the secondary analysis differs from the primary analysis, since now we're getting a little more information about the individual reads and the bases within these reads. Some of the examples that were returned by Alyssa Line and Call are shown here. This includes the per sequence quality score, which shows us the proportion of the different Q scores as a function of population, and also the adapter content that was measured to be present within each of the sequencing reads. Another metric that's provided is going to be information about duplicate reads, the number of reads that are trimmed and untrimmed, along with the information about the reads that will be present in the BAM file, the average read length, and the average read depth. Alyssa Leinenkall also performs an analysis on the coverage of our experiment. This is done in terms of the percentage of target bases and percentage of target regions, and it provides the depth for both of them in the tabular form, such as on the left, or as a graphical form on the right. In this case, we see that 99% of our target regions were covered with at least 20 reads. And you can use this information to determine if your sequencing depth was appropriate for your analysis. Alyssa Leinenkall provides a summary table for all the variants that is easy to read. This tells us the CNVs, SNPs, and translocations identified across each of the genes in our target analysis. Alyssa Leinenkall also allows us to view the read pileups for every call in the sample to verify these. The data in each sample is preserved for any future reference and is accessible wherever you have a web browser. Shown here are two true variants that we wanted to highlight. The left one is a single nucleotide deletion that we see across most reads within this example. And on the right, we have a single nucleotide variant where we had a C to be called in the place of where G was expected, and that's highlighted by blue in all the reads that are visible. In addition to that, we can see that there are some potential read artifacts that get highlighted wherever we see any sort of single variants or single examples of reads, such as the green A's on the very right of this image. This allows us to confidently know that the single nucleotide deletion and the single nucleotide variant are likely true positives, while there are some potential artifacts, and we can verify this through visual inspection. For CNVs, what this looks like is more of similar to an array profile, where we have the individual reads highlighting in log format, the potential for being a gain or deletion, along with the total call for that entire region highlighted in blue for gains and red for deletions. This is also an easy-to-read interface that can be accessed throughout the analysis stage. While we have covered the alignment and calling module for the Alyssa family, we actually have two separate modules where we have alignment and calling that covers the sequencing reads to the variants, and the interpretation module, or the tertiary module, where we have interpretation and reporting of these variants. While these are separate modules, the alignment and calling and then interpretation and reporting modules can communicate with each other. For example, the alignment and calling module can send variants and QC data through an audible sign-off process to the interpretation and reporting module. Likewise, the interpretation and reporting module can refer back and allow you to look at the read pileup and the primary and secondary analysis QC through a direct link as well. This is how our alignment and calling, Alyssa Align and Call, and our interpretation and reporting, Alyssa Reporting modules are able to work together in your one workflow. Let's dive a little bit deeper into the tertiary analysis module for Alyssa, which is Alyssa Interpret. ListInterpret helps customers automate their variant analysis by building up a pipeline that represents their workflow to identify clinically relevant variants that can be used for reporting. We call ListInterpret a white box solution, meaning that customers get to configure their workflow to fit their needs, and they also have full traceability within the workflow to know how variants are moving. Taking a brief look at how ListInterpret processes your molecular variants, CMVs, and translocations, a ListInterpret will input all of the variants that were identified from your secondary analysis pipeline and run them through a series of filters to help you decide which variants will be curated and whether or not these variants will be included in the report. A ListInterpret can help you get from a long list of variants down to just a short list that require a deep dive through the literature and clinical databases. To assist with the curation of variants, a ListInterpret provides all of that clinically relevant data on one screen for rapid variant curation. This includes information such as population frequency databases or information from clinical databases. Variants can then be included in the report and include the annotation and commentary that is provided free text by curation staff. You can also access public and premium data sources with direct link outs that will allow you to have the most up-to-date information in the databases that are being referenced by a ListInterpret. If you also have a lot of in-house data, you can leverage our managed variant database so that you can keep on annotating the same variants that you see over and over again the same way. The final step in the workflow is the report generation. A ListInterpret provides configurable, flexible reporting system for rapid report generation and updating through the variant curation step. This report is easy to read for clinicians and genetic counselors or your target audience. Reports, when they're signed off, can be in PDF, Word, JSON, or XML format so that you can import these back into your LIMS systems if desired. We also help you save time and remove the need for writing and rewriting the same report text for each variant by leveraging the internal database that you're building up within ListInterpret as you're importing cases and reporting out these variants. Now that our lung biopsy has gone on its NGS journey, the focus has really changed from just a few variants to all of the variants that can be identified through our NGS panel.
At the beginning of this sample's journey, it was positive for PD-L1 expression and was negative for all standard molecular analyses. However, a single next-generation sequencing test was able to better characterize this sample with an ALK myo16 rearrangement between chromosomes 2 and 13, along with an ERBB2 amplification. This workflow can work for anything from a few genes you are interested in to an entire exome plus UTR to address your genomic-based scientific questions. Thank you for listening to this presentation today.